Thank you everyone for coming to the second event in our Chemie virtual seminar series. I've been delighted with the response to these departmental seminar series and with the opportunity to showcase some of our most exciting recent research and our incredible faculty members. One of the things that we're going to cover today is some amazing research from Dan Anderson's lab. But first, I want to share that our first event, Cartish Mantirum's wonderful seminar on ammonia productions for fertilizers in the developing world, is now up on our website. So if you'd like to check it out, I strongly encourage you watch it when you get a chance. And Heather will share the link in the chat. With that, it's my real pleasure to introduce my colleague, Dan Anderson. Dan Anderson is a full professor in the Department of Chemical Engineering. He also holds teaching responsibilities in the Institute for Medical Engineering and Science and is a core faculty member at the Koch Institute for Cancer Research. Dan did his undergrad work at UC Santa Cruz and his PhD in molecular genetics at UC Davis under Stephen Kozlowski. He then did his postdoc with our own Bob Langer before joining the faculty in 1999. Dan's research is in the area of biomaterials and nanotherapeutics, and he is particularly interested in problems related to in vivo drug delivery. His research has resulted in the publication of over 400 papers, patents, and patent applications. He has founded 6 companies to date. Today, Dan will be talking about 2 projects from his lab, one on encapsulated cells for diabetes treatment, and the other on mRNA therapeutics for genetic diseases such as cystic fibrosis. Please join me in welcoming him.
Thank you so much, Paula. While I load my slides here. Terrific. So first, thank you so much for taking the time to listen to our story. And I'd like to tell really 2 stories from my lab that are both based on the materials we use for medical devices. And so I thought I would start off with this, which is Yorick. So Yorick was a construct created by Ed Mueller at the FDA to help educate on medical devices in the 1970s and 80s. And as you can see, Yorick has had quite a few medical procedures. In fact, he had almost all of them at that time, and was really used to show all of these different interesting devices. You can see artificial ribs, pacemakers, ocular implants, various tubings, pumps, vascular grafts. And, you know, Yorick was retired in 2003, but the field has, of course, continued to advance. And today, there are really an amazing number of different medical devices that were used. And, you know, a fundamental question when you're building a medical device is really, what do you build it out of? And just as an example of some of the early work, vascular grafts, which were created to provide blood vessels for people that were suffering injury and didn't have viable transplants. In fact, vascular grafts were made out of Dacron. So the scientists and doctors knew they needed something they could sew with. And so those first vascular grafts were Dacron. And this trend really continued in early medicine. The artificial heart. So the doctors and scientists knew they needed something that had a good flex life and was very flexible. So in fact, the first artificial heart was made of the same material as a lady's girdle, polyether urethane. And this trend really drove important advances in medicine, but it also became clear that off-the-shelf materials could be inadequate for medical application. And so, for example, this artificial heart, when blood hits the surface, it can clot, and those clots can go to the brain. And so a real revolution in the field, with much of this actually happening here at MIT, started with the rational design of biomaterials. So now instead of going to your thrift store or thinking of a material that already exists, you really sit down and actually design the perfect material for a perfect medical device and go about this in a traditional, chemical engineering way by making it, prototyping it, and characterizing it. One of the early examples of rationally designed materials are something that probably many of you have had personal experience with, biodegradable sutures. So these are sutures made out of polymeric sugars that upon exposure to water can break down and be absorbed in the body, so you don't need to go back to your doctor to have these removed. A more recent example of biodegradable sutures are shape memory biodegradable sutures, and these were developed at MIT. These are also fully biodegradable sutures, but they can be programmed to take on a shape. And so as you can see, when you shift the temperature from room temperature to body temperature, on the left, you have a suture that takes on the conformation of a screw. And on the right, we have a suture that actually ties itself into a knot. And this is just, in my mind, a beautiful visual example of how the rational design of these medical materials is going to continue to play an important role in medicine. But a fundamental challenge is that it's still quite difficult to make these things, and often it's difficult to define the perfect design criteria for a specific biomaterial. So this is just a representation of all of the steps we might imagine when we're building a new biomaterial for a medical device, and it takes quite a lot of time. You need to design it, make it, purify it, fabricate it into your device, study it, and then ultimately, of course, do animal studies, because those are often the most important data points as to whether or not this material is going to work. So one key focus of our work has been to think about how we can accelerate this process through automation and taking cues from high throughput small molecule discovery. So now, instead of sitting down and trying to make the perfect material one at a time, we try to come up with ways to make literally thousands of materials and to screen these very quickly using automated systems and also with biological systems, so that we can really learn early whether or not these have the potential to be used in people. And so the first story that I'd like to tell is about our work on a living drug factory. So this is basically a device, but instead of being a device made out of plastics and electronics and filled with an exogenous drug, it's actually a living device. So this has living cells, and these are placed in the body inside a material that protects it from the immune system, but will allow for your body to keep it alive with nutrients and oxygen, and to secrete drugs that can be important to treat the patient. And the real motivation behind this work for us was thinking about the disease diabetes, and in particular type 1 diabetes, which is a result of a patient's own immune system attacking the beta cells in their pancreas. And so these are the cells that produce insulin, which ultimately end up getting destroyed, leading to a loss of insulin production and sugar control. And so the goal is to think about how we might actually engineer a new pancreas that can resist the immune system of the patient and then restore that control of blood sugar. And the goal is also not just to make a customized pancreas for a patient, but in fact, make a device that could work in everyone. So really thinking about this as one device to treat all of these patients that have this disease, regardless of their genetic differences and immune differences. And so sitting down as an engineer, we thought, well, what are the design criteria for a device like this? Well, first, as I mentioned, this is a living device, and so it needs to be supplied with oxygen and food. And so in our bodies, of course, these cells are supplied with oxygen and food by blood vessels. However, even today, engineering complex three-dimensional blood vessels can be challenging, but it's really fundamentally important in building tissue-engineered constructs and living devices, because this is an old rule of thumb and a visual to demonstrate that. But basically, when you have a three-dimensional living piece of tissue, if it doesn't have blood vessels, once it gets to about 1/2 of a millimeter, you see death on the side of the organ that is furthest from the blood supply. And as you might imagine, the part of the organ that's close to the blood supply is getting nutrients and oxygen, but the deeper you go into that construct, there's more and more competition for those nutrients. And so you get starvation and death. And so since it's quite challenging to make these complex vascular beds, and we need to supply food to all of these cells, what might we do? Well, in fact, one idea that's driven this field now for decades has been to make them small. And so there's no reason that we need to make this new pancreas look exactly like the old one. And in fact, in my mind, the most exciting work in this field has been thinking about building many, many small pancreas. And so now instead of building a large organ with blood vessels and complex structure, we build these small spheres of water-rich gel. And what you can see here is inside of these spheres are living cells. In this case, these are pig cells producing insulin, which is why they're staining pink. So in addition to keeping these cells alive and allowing for the influx of nutrients and oxygen and hormones and insulin, we also need to protect them from the immune system, because as I mentioned, the patient's own immune system has destroyed their pancreas, and this would happen again. But in this context, we're thinking about creating a device without genetic matching. And so we also need to think about organ rejection. And so we want to protect these cells from the immune system, but allow them to function. And so we think of this in a classic chemical engineering context as creating a semi-permeable membrane around these cells. It allows what we want in and out and prevents those immune cells from interacting directly with the beta cells. And this is just a nice visual example of why we need to do this. This is a beautiful video from Eric Betzig at the NIH, who got the Nobel Prize for his imaging work. And here what we're looking at is actually a T cell. So this is an immune cell shown in orange, attacking, in this case, a cancer cell, but the principle is still the same. We need to physically separate these elements of the immune system from those therapeutic cells that we have inside this device. Otherwise, we will get this attack and ultimate destruction of the transplant. And so it turns out that these types of microcapsules really do everything that we want them to. They will allow nutrients and oxygen in, they will allow hormones out, they will prevent those T cells from interacting with the insulin producing cells inside. But the early studies as far back as 40 years showed that they would only last in animals for a couple of weeks. And so there was a mechanism by which these were getting shut off. And it turns out that, of course, our immune systems are smart. And so if our body sees a surface that it can't process and digest, it will often cover it up in scar tissue to wall it off from the rest of the body. And so in this case, when these small gels with living cells are placed in the body, the material itself is recognized as foreign and then is covered in layers of scar tissue, leading to isolation and ultimately starvation and death of the cells inside. And this has really been the fundamental challenge to getting these types of devices to work. And it's also proven to be a barrier to the application of many different materials in different medical applications. So this is in fact data from a human in one of these earlier studies from about now 12 years ago, where these small gels were placed inside the abdominal space of this patient. They had these insulin producing beta cells. And when the doctors came back, they could see that these capsules had adhered to the wall of the abdomen and become covered in layers and layers of scar tissue. And so if you have experience with the microscope, you might recognize here is your sphere. Here are the cells inside that are supposed to be producing insulin. And this is the layer and layer of collagenous scar tissue that has led to the death of those cells. So the fundamental next step at this stage was really thinking about how might we make a better material. And so now I'm going to dive into a little bit of chemistry and chemical engineering for those of you with a history here at MIT. So this is the chemical structure of a polymeric sugar called alginate. And really, this is the material that's been looked at the most in the field for decades. And we reasoned that this material has a lot of properties that we like, and it has some properties that we don't like. What we like about it is it's very hydrophilic, and so it allows for passage of water and water solubility. Importantly, it also has this ability to cross-link and form gels in the presence of calcium in a way that is very cell-friendly. So as you can imagine, you can make a solution of this polymer, drop it into a little bit of calcium, and it will form a gel. All of this in a very gentle way that allows for the cells to be undamaged during the process. And so this is an example of how this works. You take your alginate, which is derived from seaweed. Actually, not this type of seaweed. That is the seaweed from a Japanese restaurant, but a related form. It's processed through a droplet generator to make these precise droplets, dropped into a bath of calcium. And when these droplets contain cells, they cross-link into these beautiful little mini pancreas that I showed you earlier. And so this is a slide showing certainly over 10 years of work by 10 to 15 people, probably more actually. And I don't want to dive into all of the details, but just give you a sense of the workflow and process that we engaged in to try to solve this problem. So first, we spent time thinking about what types of chemical modifications we would add to this alginate polymer and how we would do the chemistry. We developed automated systems so that robotically we could make these things much faster than we could by hand. We then would characterize these using conventional chemical methods, form droplets and spheres, and then immediately look at their ability to elicit an immune response in animals. And so we made hundreds of polymers, we turned them into gels, and we looked at whether or not they stimulated the immune system. Those materials that did not lead to immune activation, we then went on to advance into further studies in rodents to see whether or not they could resist the scar tissue formation, to see whether or not they could be used to actually cure diabetic mice when they had islets, and then ultimately moved on to primates. And so here's one example of a workflow once we had identified lead materials for this, working with my colleague Doug Melton, who has created a way to take embryonic stem cells or induced pluripotent stem cells and turn those into human beta cells through a multi-step controlled process. We were able to take these human cells that he had made basically in a reactor. So you can imagine a limitless supply of these beta producing cells. We put them through our droplet generator. We made these small gels and then implanted them into mice that had a strong immune system. And so this is actually a data slide showing the blood sugar of these diabetic animals over the course of the experiment. And so on day 0, you'll see this blood sugar is very high because the animals are diabetic. They don't have control of their blood sugar. After implantation, we see that the blood sugar drops to the normal range. And then for as long as we did the experiment, that blood sugar control was maintained. And so this was an important demonstration that these devices could actually allow human beta cells to work in diabetic rodents and affect their disease state for an extended period of time. We went on to advance this into non-human primates, where we actually took the beta cells from one primate, we placed it into our gel capsules, and then we placed it into a second primate to see if we could get survival and protection of those cells. And so this is actually some microscopy images of the different devices. This is what they look pre-transplantation. So you can see here, for example, is a gel capsule. Here is a primate islet on the left side. And after 4 weeks, when you use the conventional unmodified materials that people had been using before our work, when you come back, you see just what people had seen in humans. These are covered in a layer of scar tissue, and the cells are dead. However, with the new MIT materials, you could put them in and then come back a month later or even 4 months later, and the cells are alive and functioning. The capsules are clear from scar tissue. And this really gave us hope that this was not just a tool we might use in the lab, to manipulate the biology of mice, but in fact had real potential to be used as a therapeutic. And so in 2018, so we actually had started a company called Siglon to advance this. This is now a public company. And in 2018, they announced that they had developed a strategic collaboration with Eli Lilly to advance this for the treatment of diabetes. But we view this as a technology that's not just for diabetes. In fact, this is a platform for making drugs for a range of different diseases. And so when we think about this technology, first we think about the cell. This is the factory of the drug that we're trying to make. So we can program the cell to secrete all kinds of different factors, whether they're for the treatment of hemophilia, Fabry, MPS, all of these different genetic diseases, and of course diabetes. We then have created this capsule that protects those cells and allows them to function as a drug-producing reactor in the body. We've identified where in humans we want to place these so that they can maintain their function long-term. And ultimately, the goal is, in fact, to have this as a long-term solution for a range of different diseases. That these are going to be producing the drugs we need on demand for as long as we need them. Excuse me. So this is the clinical status of the technology at Sigilon. In fact, phase 1 studies have actually started just now for hemophilia. In fact, it's too early to even talk about them other than they've started. And we're excited to see how this technology progresses for diabetes and also for other diseases. So now, excuse me, I'm going to switch gears and perhaps go a little bit faster and end on this topic. So another aspect of what we're interested in doing is actually making drugs that can repair your DNA while you're still using it. So imagine, for example, if you had a liver disease, which is why I've shown this liver in green, it could also be a lung disease, or it could be a disease of the vascular system. Could we create a drug, or in fact a nanoparticle drug, that could be injected into your body, travel to those disease cells, actually enter them, specifically repair this genetic defect out of all 3 billion base pairs that you've got in your cell, and then ultimately lead to a restoration of function? So, actually this was the kind of thing that motivated me to come to MIT way back in 1999. And at that time, really sounded like science fiction, but I think there's growing evidence that in fact this really is going to happen. And I think it's going to have a big impact in medicine in the near future. The challenge is, of course, how might we do this? So we envision building nanoparticles that can deliver these genome editing tools inside of cells. And as you could imagine, it's not an easy problem to solve. So if we try to break down all of the steps we imagine as challenges, first we need to complex these tools inside of a nanoparticle. We need to be able to have this nanoparticle travel through the bloodstream, avoiding off-target tissues, reach the destination we're trying to treat, and then leave the bloodstream. Then they need to actually even enter the cells. And so this is typically thought of as a process of cellular eating or endocytosis. So these therapeutic nanoparticles are eaten by the cells, but then once they're in a cell, they're actually in a cellular stomach, and they need to escape that. And so we need to engineer these particles to escape the cellular stomach. They need to then realize they're in a cell and release. And ultimately these payloads need to reach, in the case of genome editing, the nucleus so that they can interact with our genome and specifically repair defects. So it sounds like an amazing set of challenges, but in fact, there's been enormous progress. And so where we've started to see the most progress is that there are organs that are naturally amenable to nanotherapy. So in fact, for a range of different chemistries, if you inject nanoparticles in the blood, they will often end up accumulating in organs that play a role in filtering the blood. So these are organs like the liver, the spleen, the bone marrow, immune cells. But getting these nanoparticles to those cells is not the same thing as getting therapeutic function in those cells. So how are we actually going to get inside of the cells in vivo? Well, maybe just first a refresher. Cell biology is, in fact, controlled by nucleic acids. I think everybody's heard of DNA and RNA. And these are really the fundamental drivers in molecular biology of the cell, and really provide the information content that programs what our cells do and also what diseases we face. And so what we've started to see already is that intracellular delivery of nucleic acids is revolutionizing medicine. And the fundamental challenge is not, do nucleic acids work, but actually how do we get these nucleic acids inside of cells that we want to treat? So how do we deliver them? And unfortunately, nucleic acids are simply not good drugs. And what I mean by that is when you look at the properties of a drug, that you might take in a pill, it has certain chemical properties that allow it to reach cells without processing and damage. But RNA or DNA they're large molecules, they're highly charged, they don't cross cellular membranes, they don't naturally get eaten by cells. They are prone to being chopped up into little pieces by nucleases in our bodies. And we can even recognize them to induce immune responses, which in many cases can be negative. But there's been huge progress. And these are really the three keys to making an RNA drug. The first is sequence selection. So a lot has been learned about how you can actually program the RNA to have it potent and specific. Chemical modification, so changing the chemistry of the nucleic acid, whether it's RNA or DNA, to give it drug-like properties and facilitate delivery. And then encapsulation, building a nanoparticle. And this has, in fact, been a key driver in enabling this technology today. So, as is the case for our living devices, again, we're faced with this question. If we're going to build a delivery nanoparticle, what material are we going to use to build it out of? And I thought at this moment I might digress to say, well, perhaps some of you already have personal experience with RNA lipid nanoparticles. I know I am very anxious to get injected with some, since today these COVID vaccines are in fact RNA lipid nanoparticles. And this is just a news release talking about how good they work. They really are amazingly effective drugs. And interestingly, when you look at the chemistry of these drugs, they really look a lot like all of the nanoparticles that we've been working on for quite a while to facilitate genome editing and other types of RNA therapy. So they really have the same basic properties. And again, to dive into a little bit of chemistry, for those of you that are hungry for that, this is the composition of your canonical RNA lipid nanoparticle. So it has RNA, but it also tends to have 4 different types of lipids. The first and most important is what we call the ionizable lipids. So in the upper right, I've shown the structure of one developed in my lab. This is an amine-containing lipid that can interact through charge interactions with the RNA to form a nanoparticle. And in addition to this, it plays an important role in escaping the cellular stomach by facilitating interaction with the stomach membrane and escape. These RNA nanoparticles typically have other what we call helper lipids. So these are phospholipids and cholesterol. These are things that play a role in the stability of the nanoparticle, and also a polyethylene glycol lipid, which plays a role, again, in colloidal stability and in facilitating the circulation of these nanoparticles for long enough to reach the organs we're trying to treat. So all of these can be combined with RNA under simple mixing conditions to form nanoparticles less than about 100 nanometers. And when we first started this work, we thought, well, what is the best amine lipid to use? And at that time, really the public space didn't have that much chemistry, meaning really only the sort of 40 or 50 structures had been described. Here are some of them. And the diversity wasn't that high. So for example, the structure in the upper left corner, DOTMA, this is a lipid from the 1980s. D-lin-DMA, the lipid on the upper right versus the upper left, excuse me, really only different by a few atoms. And that was actually developed and published by a company called Protiva in 2005 or 6. So really only different by a few atoms. So the first fundamental question is, how do we increase the diversity of chemical space that we explore for RNA lipids? And so the first question is really a chemistry question. And for any of you that have been doing organic chemistry, you might say, well, look, it's not that tough for me to make this lipid. And you would be right. In fact, this is an example chemical synthesis for a cationic lipid. But if you wanted to do this thousands of times, and each time you did it, you had to make it in a customized way, both synthetically, but also thinking about the purification steps, it then becomes enormously challenging. And so our lab has focused in particular on developing one-step chemical procedures that allow us to make large libraries of delivery materials for nucleic acids This is something called a Michael type addition, and I won't dwell on it. But the bottom line is developing first a simple chemical procedure that we can use in a parallel setting to make thousands of different delivery reagents allows us to make thousands of different nanoparticles and then quickly identify those that are going to have potential as therapeutics. So now a quick refresher. Some of you may have been taught that the central dogma of molecular biology is that DNA encodes for messenger RNA, and that messenger RNA encodes for proteins. And so there is this, of course, exciting field now thinking about using messenger RNA as a drug. So we will deliver exogenous messenger RNA inside of these cells. This will then be turned into the proteins we want, whether they're spike protein for COVID or cystic fibrosis treatment. So there's no question that messenger RNA will be turned into protein inside of cells. The question is, again, how do we deliver it? And I want to just give very brief examples. Here's an example of nanoparticles developed in our lab used in an animal. And these are nanoparticles encoding messenger RNA from fireflies, luciferase. And so what that means is actually when you inject mice with these nanoparticles, their tissue will actually glow, give off light that you can measure. And so you can figure out where your nanoparticle works, if it works, by looking for light coming out of these animals. And so here I'm showing two different nanoparticles, one that goes to the lung and one that goes to the liver. And so in this experiment, you can see that one of the nanoparticles is making the lungs glow because they've delivered messenger RNA inside the lungs. However, that same nanoparticle does not give any signal in the liver. And then the opposite is true with the other nanoparticle. The lungs really don't give off much signal, but now the liver does. And so a big part of what we do is try to develop tissue-specific nanoparticles that can allow for expression in different tissues. A really interesting way to think about using nanoparticles for the lung is actually not via injection, but actually by nebulization, so breathing them in. So this is an old-fashioned way to deliver therapeutics to patients. You can actually make these nanoparticles in solution and then subject them to a vibrating mesh, which makes a mist of small water droplets. And this mist can be breathed in. And when we do this with messenger RNA nanoparticles, what we see is really high transfection of the lung. So the epithelium of the lung, and really no expression in other tissues. So this is an exciting way to think about relatively non-invasive messenger RNA therapy for the lining of the lung. And a collaborator of ours, Translate Bio, has actually moved this forward for the treatment of cystic fibrosis. So cystic fibrosis is the most common fatal inherited disease in the US, with over 30,000 patients in the US. And basically the median age of death is about 30, and really compromises the quality of life. And in particular in the lung, where patients essentially lose the ability to breathe. So the solution is to build a messenger RNA nanoparticle that can be nebulized, breathed in, would be self-administered weekly, and would actually deliver a complete, correct copy of this CFTR gene to patients. So what that means, some patients have a version of cystic fibrosis that actually can be treated with small molecules, but some patients have mutations that really are untreatable. And so this is a therapy that really, independent of the mutation of the patient, is going to be useful, we hope, for treating everybody. And again, the way it works is you breathe this in. This is a nanoparticle with a messenger RNA encoding for the cystic fibrosis transporter. It will enter these lung cells, degrade, release the messenger RNA that will then get turned into protein and then expressed on the surface of the lung cell so that it can allow for ion transport and proper function of the lung. And in 2019, they announced the interim results of their first study. And excitingly, not only was it clearly safe, but it resulted in a marked increase in FEV, which is a measure of lung function in these patients at a single dose. And so they are currently looking at multi-dose studies right now. I think this is a very exciting potential treatment for the disease. But I'm also excited by other ways nanoparticles can be used to treat this. And I realize I'm a little short on time, so I'll go quickly. As I mentioned, we want to actually permanently correct genetic disease. And we think we can do that with lipid nanoparticles. The trick to getting this to work is actually to take advantage of your own DNA repair pathways. So, as you might expect, as we go through life, we're experiencing insults to our DNA, whether it's sitting out in the sun at the beach or getting an X-ray. There are many different types of DNA damage we can face, and we have mechanisms for dealing with these. But we can actually take advantage of these endogenous mechanisms to trigger them to do therapeutic events. And there's a lot of different ways to do this. I'm only going to talk about one, which is through Crispr. So you may have heard 2020, the Nobel Prize was awarded for Crispr genome editing. This is an enzyme that can bind DNA, and it can be programmed to specifically cut at therapeutic sites. And once you create a cut, you're actually activating these repair pathways in a way that can be therapeutic. And the way you program this is by delivering what's called a guide RNA. So that's shown here. This guide RNA actually targets it to this specific cut site. So I should probably go quickly, but basically the way this works is you have the Cas9 protein, you have the guide RNA to target a cut where you want. And if you do this, you can specifically turn off genes permanently. And I think in the interest of time, I'm going to go to the punchline, which is that we've been able to make nanoparticles that can do this. And now companies have started to take this to the clinic. And so here, for example, is primate data from a company called Intelia. And what you're looking at is the level of the target gene, which in this case was TTR, which I could talk about later. And after a single treatment in primates, they can basically completely knock it out. And they have recently announced the start of human trials using these nanoparticles. And we're optimistic that these can be used not just for this disease TTR, but for a range of genetic diseases. And the hope is that we can actually build therapies that you might take once that could provide long-time cure for patients. And I thought I would end also that, to me, this idea of building customized materials isn't really limited to living devices and nanoparticles. We think building smart biomaterials has the potential to impact many different disease states for a range of applications. And we've started several companies to help move this stuff forward. I'll probably end here, and I'm happy to answer any questions.
Thank you, Dan, for a great seminar. It's really exciting to hear the advances in your work and also the translation. We already have a question from David Carroll. Seems like you're making artificial viruses, is the question.
Well, it's an interesting way to put it. I mean, you know, really some of the most exciting gene therapies today are viruses that have been reprogrammed. However, you know, those viruses today also come with limitations. Perhaps the most important of which is that you can only dose a patient once before they generate an immune response to that. So you better get it right when you treat them. We are trying to make, in essence, nanoparticles that can do this delivery piece of what a virus, a program virus, can do. But also, because they're fully synthetic, there's absolutely no risk that they will do anything we don't want them to do. At least that's the hope.
Excellent. Mark Harvey has a question. Could you create a nanoparticle that would deliver CFTR to multiple organs at the same time?
It's a great question. So cystic fibrosis, I think most people know about its disease impact in the lung, but it also does impact other organs. And there certainly is exciting data indicating that nanoparticles can be made to reach a range of tissues. I would say today, there is no messenger RNA nanoparticle that can hit every cell in our body, but there are messenger RNA particles that can hit different cells, whether they're the lung, the liver, the endothelium, immune cells, various local applications, muscle. And so the list keeps growing, and there are a lot of smart people working on this. So I am optimistic that we will see nanoparticles that can more broadly provide therapy to patients.
Thank you. Josh Reigelhaupt asks, how much diversity is in the nanoparticle world? Yesterday, Beam did a deal with Guide Therapeutics. What is their platform? How is that different than your efforts? And can you describe the various generations of the nanoparticles and what today's challenges are?
That could be a long answer. So I'd start actually, Guide Therapeutics was formed by James Dahlman, who was my graduate student. And when he was here at MIT, what we had done was develop barcoded systems to allow for multiplex testing of nanoparticles. And so what that means is that instead of making one nanoparticle, testing it in an animal and finding out whether or not it works, you can make hundreds of nanoparticles, try all of them at the same time, and then pick out which ones work. And so it incredibly accelerates the ability to get animal data. And this is particularly important when you think about using primates. And so he went on to become a professor at Georgia Tech. And I was happy to see that they formed this company that did a deal with Beam. And so, the key strength of what they, again, I wouldn't want to speak for them. I don't have a relationship with them, but what's great about what he's done is to leverage this multiplex system to get in vivo data on many, many different nanoparticles. And of course, we also have been doing this work with a variety of people and companies. And I think the lesson is that you really don't know if nanoparticles work until you get them into higher species. I mean, mouse data is important, but primate data is really important. And so anything we can do to accelerate that is important. And a lot has been learned. I mean, as I said, there's a growing list of different nanoparticles that can treat different parts of the body. And in fact, my mom was just injected with a messenger RNA nanoparticle, right, for COVID. So the list is going to grow. I think this is just a start.
Perfect. We have a question from Joyce Lynn. How long do you expect those encapsulated cells to last? Is there a half-life?
Also a great question. So, the short answer is we don't know. So what we do know is for the duration of the experiments we've done, we can get therapy well over a year. But our hope is that this could be something that lasts over a decade. So there are patients who've had islet cell therapy with immune suppression. So this is no polymers, but just the cells. And some of these patients, it's 13 years later, they're still not taking insulin. So that gives us hope that these things could actually provide very long-term therapy, but time will tell.
That's really compelling, actually. We have a question from Arthur Lee, Chevron Fellow. What kind of manufacturing process at scale would be used to manufacture the nanoparticles?
Well, this sure is in the news a lot now. I think, companies like Moderna are targeting, 1 billion doses or more. In fact, Paula is on the scientific advisory board of Moderna. Maybe you should ask her. But, from my perspective, this is the kind of question that really requires input from the companies. At universities, we really try to do the early innovation and development. And I do have interactions with companies where I do see some of the later stages. But when we think about these billion dose scale, this is really company-based level engineering. And all I can say is clearly by the hundreds of millions of doses that people are making, it can be done.
Perfect. Jim Bray has a question. Is there any potential to use your DNA nanoparticles to correct the long-term lung damage associated with some COVID cases?
Oh boy. Yeah, well, it's an interesting question. I think I would need to know more about what that damage is. I think, I guess the one way I would answer it is that there is evidence that these types of therapies can have an impact on fibrosis. And I know that is one piece the type of damage that people see in the lung. There's data in the liver actually with certain nanoparticles showing that you can deliver to stellate cells and get an effect on liver fibrosis, although it's still quite a challenge. So the short answer is I don't know, but I certainly would hope so.
Yeah, I think that's a really exciting area, actually. And here's one that I think is very relevant today. Do you see an immune response to the LNP, and can you do repeated dosing?
Yeah, it's a good question. Of course, you know, the nanoparticles we're all hoping to get soon are designed to give us an immune response. And so certainly you can create them to give an immune response. And you can also create nanoparticles that don't give an immune response, both by engineering the RNA the right way and also the lipid structure the right way. And there are a variety of studies showing that repeat dosing is possible. In fact, I didn't have time, but the first RNA lipid nanoparticle drug was actually a drug called Onpattro, developed by Alnylam and approved by the FDA in 2018. This is a drug that patients take every 3 weeks for the rest of their life. And it has really been amazingly useful, and so far has demonstrated the ability to have repeat dosing.
Thanks, Dan. We have a question from Partha Ghosh. Are there any ethical questions we have to deal with as we advance mRNA, start fixing basic constructs of the human system? Looks like it says, can engineering and spiritual engineering, can chemi engineering and spiritual engineering converge?
Huh.
Or must they converge?
Well, you know, certainly CRISPR-based genome engineering has resulted in a lot of discussion on this point. And I would say it's certainly an area of active discussion. When is it appropriate to do genome engineering? When is it inappropriate? And I, as I sit here today, wouldn't want to be the person that draws that line. But what I would say is there absolutely are cases where it's completely appropriate. And just for example, I'm a founder of a company called Crispr Therapeutics. They've already shown the potential for using CRISPR to treat patients with hemoglobinopathies like beta thalassemia or sickle cell. And these are horrible diseases that really don't have good solutions. And so the fact that they use genome engineering to give these patients relief, in my mind, is completely appropriate. I recognize the challenges with what some might think of sort of optional use of CRISPR. And I think it's an important area for continued discussion.
All right. Gabriel Abgarinos has a question. Can the mRNA particles also be used to repair damage from NTM in the lung, non-tuberculosis mycobacteria?
Yeah, again, I would need to know more about the type of damage caused by these diseases. But you know, I would say most of what we think of today is alleviating genetic disease or interfacing with the immune system to create therapies. So I could imagine that it could be used to cease destruction. But again, I'd have to know more about the type of damage to understand if it could be therapeutic.
The next question is from Joe Kuhach. It seems nanoparticles work in organs that filter, liver, kidneys. What about heart and coronary artery disease?
Yeah, well, so great questions. I think there is a growing list of evidence that the endothelium can be accessible with nanoparticles. And so we've shown that in a variety of systems and also in primates. So obviously, if you can affect the endothelium, you might imagine using that to treat cardiovascular disease. There are genetic therapies that may be useful in the heart. You can also imagine, you know, it might not just be via intravenous injection. There are people looking at injection directly into the heart muscle as possible ways to deliver to the heart. And again, there is proof of principle and some clinical study ongoing.
I think we have time for one last question from Tom Neal. Can germ cells be changed, and are ethical considerations an issue?
Well, so germ cells have been changed. You know, there is this very famous study with CRISPR. This is not with nanoparticles. This is sort of baby engineering. And this is absolutely an area where there's very vigorous ethical discussion. Can a nanoparticle be used to affect germ cells in patients? To my knowledge, I'm not aware of one that does. That doesn't mean it's impossible. And I'd also say there may be diseases where that could be appropriate. But again, I think you start to approach these very difficult ethical conversations, and it's going to take some real discussion.
Thank you, Dan. It's clear that along with the excitement and the cures that are coming from these technologies, it gives us an opportunity to think about how we use the tools in the future. But huge amounts of opportunity. And we're really excited about your work. I think everyone thanks you for giving us this seminar and also such a wonderful discussion. I think there were so many insightful questions and wonderful answers that you were able to provide. So let's all thank Dan. And I know it's we're not in an auditorium, but we show our appreciation. Thank you very much, Dan.
Thank you so much. I enjoyed it. Thank you.
And a reminder that this will be posted and it will be closed captioned in the posting on the website once we're able to get it up. Thank you, everyone. It's been wonderful to connect with you one more time. We love our alum and we love your enthusiasm and interest in the work that's going on here at MIT ChemE. Thank you.
Thank you. Bye-bye.